Hi, everyone. We're pleased to have you all join us. I'm Lauren Costi, Scientific Account Manager at CDD, and I'd like to welcome you all to our Collaborative Drug Discovery Quarterly Product Update webinar focused on our automation and inventory management capabilities. So before we dive into the exciting updates we have for you, I'd like to quickly go over a few housekeeping items. For any product-specific questions, please use the Q&A tab in the toolbar. This will help us address your inquiries efficiently during our dedicated Q&A session, as well as go over any quick ones as we go. For all other questions, comments, or general discussion, please use the regular chat box tab in the toolbar. We encourage your active participation throughout this webinar. If you would like to request a one-on-one customized demo for any of these features, we'll have a poll to easily request one before the Q&A segment. So today's updates. For automation tools, we're going to go over enhanced parser interface for data standardization, new delete samples API endpoint, and streamlined CRO data integration. In regards to inventory management, we'll go over automated alert system for thresholds and expiration, unorganized location management, enhanced search with batch identifiers, and automated sample ID generation. These updates aim to streamline your lab operations and CRO collaborations. However, before we get into those, if you're interested in AI and curves, I recommend you check out the Q4 product update webinar. Curves received improvements such as area under curve calculations, replicate point visualization, bell-shaped curve fitting, and color coding logic updates. In regards to AI integration, we went over similarity searching in chemical databases, bisoiosteric suggestions, QSAR API access, and our AI support chat box. For detailed information about curves and AI updates from our Q4 2024 release, a link will be provided in a chat and can be found on our website as well. These modules have been developed based on user feedback and research requirements with the goal of streamlining your analytical workflows and accelerating discovery processes. Your feedback continues to be invaluable to us, so we encourage you to engage actively throughout this presentation. Thank you again for joining us today, and let's begin our product update journey. We're going to start with automation tools.
Hi everyone, I'm Kaylee Anderson, the Director of Business Development for the Southeast US here at Collaborative Drug Discovery. Today, I'm going to walk through 3 key areas where we've enhanced the automation module's functionality. For teams managing CRO collaborations, these updates streamline your entire data processing workflow, from initial data receipt through analysis. First, we've expanded our parser capabilities. That means the new module now includes assignment management with undo, redo functionality, enhanced mapping template creation, and automated calculation setup. Second, we've improved API integration. You can now programmatically import and process data, execute pre-configured calculations, and monitor data integrity in real time. Third, we've streamlined CRO collaboration. This means you can configure templates for repeated use, process and validate data in real time, and generate standardized results automatically. You can set up your templates and calculations once, then use our API to automatically process incoming data through your established pipeline. This ensures consistency and saves significant time in routine data processing. And now I'm going to hand it over to Pranitha, who'll demonstrate these features in action.
Hello, everyone. Thank you for joining today's webinar. My name is Pranitha, and today I would like to guide you through how you can automate your data processing workflows from instrument integration to results generation. We will focus on 4 main steps in the configuration process. First, how you can define your experimental protocols to hold your assay data, transform your raw instrument data, and save your custom parser for further use, streamlining data mapping for consistency and automation, and integrating your instrument data directly into the system. Here is a report or data table that shows an example of an assay that has been tested against a few molecules against different commercial cell lines. What you see here is IC50 calculated over different cell lines, and this is a live dose response curve with the run date here and the cell line. So these are the results we're looking for. So let's see how we can set up a protocol to get these results. So protocols are just containers for your assay data. It not only stores your data, but performs calculations over it. Once you create a protocol and its framework, you don't need to redo it every time there is new data. Instead, you can upload a new run or experiment to the existing assay and apply the same framework over all data uploads. I've set up this assay here, which shows a cell line as a condition, the concentration as a numeric data field. I have normalized the signal against positive and negative controls. We then take this normalized data and fit it against the Hill equation and calculate any intercepts like the IC50. Now that we've set up a protocol, let's go ahead and set up our raw instrument data to be converted into a tall and skinny format. You can upload the file, pick a project, and preview the file. And what you will see is there is multiple worksheets in this Excel sheet, and you have the cell line as a fixed value that you can pick. You can also have the plate name. Maybe you pick this as a tabular data, but then you realize you do not want to do it. So you also have the undo functionality. It is smart enough to look for the plate blocks, and it found this compound ID plate block. It also found another plate map here. At the end, you see there is a preview import table. So it has automatically taken what you assigned and converted it into a tall and skinny format. If you like all the assignments you have and you think you're going to save this parser, you can save it. You can even overwrite it. I will call it something to this effect. And then you can just hit proceed, and you will be taken to the mapping that you're used to seeing. You just have to pick the actual assay that we were going to do, the endpoint assay. In the cell line, create a new run, hit apply. We had the plate name come through the fixed value, if you remember from the parser. We also got the format from a fixed value, and we got the well location. And here is the concentration. And let's pick the RLU. And once you've done this, you can save this mapping as a template. You can call it however you want, whatever makes sense. And you can process the file. Here are the different parsers we have made. You can have different parsers based on the instrument they come from or from the different CROs. You can also list all your templates here based on how they were made and what makes sense to you. Now that we have set up our parsers and the mapping templates on the front end, we can use our API to actually find the parsers. I have made a call to our vault and found this is the parsers that we just made. This is the ID, you get the name and when it was created and when it was modified and who the owner is of the parser. You can do the same thing with the mapping templates. It's important to note the ID and the name so we can use it. And then using either the ID or the name, you can upload a file to the assay data using the custom parser ID and the mapping template ID. I have this file that I'm going to now upload. And here you can see that we have two runs. This was the run that we created when we set up the parsing template and the mapping template. And this is the run that we got through the API. Let's take this a step further and look at this folder. Maybe this is a folder on a SharePoint somewhere where the instrument can actually put in its output. This is the raw data that we saw, and we have already created the parsers and the templates. So we know that we have to have a file watcher for this folder, and using the same API calls, just using the parser, the Vault ID, the mapping ID, and the project ID, I will now run this small program that I have now written. And let's see. Here you see we have 10 runs. This was the run we created while setting up the parser and the mapping template. This is the run that was made through Postman, but everything else is from the folder watcher that we simply created. And now we can just look for the readouts for this assay, and you will see we have the results as expected. We have the dose response curves, they're live, they're colored, and we have everything as we need. When you don't have a workflow set up, you have to wrangle the raw data file one at a time, and this is very error-prone, tedious, and time-consuming. Once you have set up a workflow, you can now manually upload the data, and within seconds, you will have the results in real time. You can now take it one step further with the API integration. You can read the data from the instruments using the API. It'll go through the parsing and the mapping workflow that we have set up, and you can then see the results either inside Vault, or if you have any kind of downstream integration like a data lake or dashboard, you can see it through that. Automation facilitates seamless connectivity with our network of partners, enabling the creation of comprehensive workflows and placing CDD at the core as a centralized, single source of truth for scientific data management. By streamlining data processes, automation improves efficiency, reduces human error, and ensures consistent, reliable results across all projects. Additionally, it enhances scalability, allowing organizations to manage large volumes of data from multiple instruments and vendors with greater speed and accuracy. Automation ensures that data is managed in a way that aligns with FAIR principles, making it easier to share, repurpose, and collaborate efficiently across systems and teams. I hope that this session has helped you understand how to automate your data processing workflows. If you would like a demo or want to learn more, please vote so on the survey later on. Thank you for your time.
Hello, everyone. My name is Jean Geesler, Director of New Business within Canada and the Midwest of the US at CDD. Today, I'm presenting on our enhanced inventory module. So for laboratory managers and for researchers managing sample collections, these updates introduce comprehensive inventory control capabilities directly within CDD Vault. This eliminates the need for separate sample tracking or inventory management systems. So let me walk you through three key areas where we've enhanced the module's functionality. Firstly, we've implemented smart alerts, and this allows you to do threshold monitoring for critical sample quantities, expiration date tracking with automated notifications, customizable alert parameters by unit of measurement, as well as proactive inventory management triggers. Secondly, we've streamlined location management where you can now assign samples to unorganized locations, create flexible storage hierarchies, set optional capacity limits, and even track locations across different sites. And finally, and perhaps most importantly, a key area which has been updated in our inventory module would be the enhanced search capabilities, where you can now do structure lookup using unique batch identifiers, list-based filtering with autocomplete, enhanced metadata indexing, and direct navigation from search results to inventory details. So what this all means in practice is that you can now manage your complete sample inventory lifecycle from receipt all the way through to depletion all within CDD Vault. And this ensures accurate tracking while maintaining accessibility across your organization. So now I'll hand over to Mitchell, who will demonstrate these features in action with a particular focus on alert configuration, location management, and our enhanced search capabilities. So thank you, everyone, and over to you, Mitchell.
Hi, everybody. My name is Mitchell Buckley. I am an application scientist here at CDD Vault, and today I will be stepping into the shoes of a lab operations manager working on a medicinal chemistry team to demonstrate some of the new features of CDD Vault's inventory module, and really outlining how these new features can streamline your lab operations as well as increase your data integrity. Now, speaking to the idea of data integrity, the first feature that I would like to highlight is the inventory expiration alert. So I've navigated to my settings tab, my user alerts page here. Now, simply configuring a new alert here, I can add an inventory expiration alert. I can configure this to either be for a specific project or for all of my projects as I've selected here. And I can configure the frequency and the format, of course. Now, within the inventory section here, I can define the time period that I would like to be notified prior to my sample expiring. So in my case, I would like to be notified 1 month so I can plan those relevant upcoming experiments. And so this is, of course, really helpful for things like DMSO stock solutions, or maybe some of your more unstable compounds. The other alert type that we now offer is the inventory current amount. And within this case, here under the inventory section, I can select the amount in which I would like to be notified. So for my medicinal chemistry team working on a small scale, I can select less than 5 milligrams. And so whenever some of my inventories are running low, I will be notified via email. And this is really great for increasing your decision making, ensuring that you're allocating resources to resynthesize compounds or to allocate resources to synthesize new compounds. Now, in addition to these alerts, we do also now, after a lot of requests, offer the auto generation of unique sample identifiers. So in my next tab over here, under the Vault settings and sample inventory fields, you can see that I have configured a barcode sample field. I've toggled this to be must unique, and I've also toggled on the sample identifier and selected the new auto generation functionality here. And so I've chosen a prefix of S to represent sample, but of course, many users may have different prefixes that they would like to use here. Moving over to this samples overview page for a compound within my vault, you can see that I do have a few samples of this compound. And of course, I do have my barcode that's been auto-generated, and I do have my expiration date. And of course, I have been notified that this sample is at 2 milligrams via email. Now, when creating a new sample, you'll work through the traditional workflow as you have been accustomed to now for setting up a sample within CDD Vault. Of course, we can select the expiration date, maybe 1 year from now. And you'll notice here that the sample barcode identifier has been auto-generated. Now, this auto-generated barcode could be implemented into an external barcoding device where you could physically label your inventory within your lab. So this is a really exciting feature for us to be deploying. Additionally, on the location side, we can now select unorganized locations. So in today's world of a very global collaborative environment where many industries or even academics are leveraging the convenience and agility of CROs, we can track our inventory at a very general level. So here, I'm creating a new sample in just the Shanghai location for my chemistry CRO group. And as your inventory scales and your company scales, being able to conduct powerful queries across your inventory system is, of course, paramount. So one of the first updates that we've made on this search page here will be within the structure lookup button, where we can now search by any unique batch field. So for myself, I'll type in a code that my CRO has used. This is my unique external batch identifier. The structure will be populated. And now I can look at all of the inventory information about this specific compound. Additionally, we have enhanced the search, sort, and filter capabilities for synonym, formula weight, and exact mass. So I can simply add a synonym filter, for example, and I can search by any of the synonyms within my vault. So maybe I want to search for diazepam and look at all of the different inventory information for this molecule. Additionally, we can reset the filters here, and maybe I want to see all of the information for my compounds that have a formula weight from, let's say, 100 grams per mole to 300 grams per mole. I will be returned all of the compounds that fit this criteria. Another great feature that we now offer is the ability to conduct list-based searching for fields such as molecule name or molecule batch ID as seen here. Within this molecule batch ID filter, I have pasted in a list of IDs, but maybe I want to manually type in an ID, and you'll see that I'm returned a drop-down menu of auto-complete suggestions. Additionally, here we can search by the location tree that we have built within our vault. So within this drop-down menu, I can search for these IDs across any of the locations, even into the specific boxes within my inventory system. Now, when I do execute this search, of course, I am returned those 3 samples at this location. So this is a very powerful tool to maybe prep for an upcoming experiment. If you have a list of IDs that you need to test in that experiment, you can quickly identify which compounds you may have on hand in a certain location. The last feature that I would like to touch on is that we've now given users the ability to toggle on and off an inventory summary within the search results table. And so this is a really fantastic feature for seeing what material you may have on hand after conducting some type of data analysis and you're planning your next studies. So as you can see within this table, we do have an inventory summary at the molecule level, as well as the batch level, where you can see I have a link out to the specific inventory sample for this batch of compound. So with that, I do thank you for your time, and I look forward to answering any questions.
Thank you for a great demonstration of our enhanced platform features, and a big thank you to all of our speakers today for their excellent presentations on our latest updates to inventory and automation. And I'd also like to thank all of you in the audience for your attention and engagement throughout this webinar. Your interest and participation are what drive us to continuously improve our products. So before we move on to our Q&A session, I'd like to draw your attention to a poll that's now appearing on your screen. If any of these module add-ons we've discussed today look interesting to you, please take a moment to vote. You can indicate whether you'd like a customized demo or if you'd prefer us to send you some resources to learn more about these module add-ons. Your feedback here will help us tailor our follow-ups to best meet your needs. So now let's transition to our Q&A panel. We've received some great questions in the Q&A box throughout the presentation, and we're excited to address them. For those who haven't had a chance to ask questions yet, feel free to submit them now in the Q&A tab. We'll do our best to answer as many as possible in the time that we have remaining. So let's begin with our first question.